So let me introduce this unusual format of the Kanizsa lecture. As you probably know, the Kanizsa lecture is a public event, a public lecture organized by the University of Trieste, promoted by the University of Trieste since 1993, the year in which Kanizsa died. At the time, we thought that having every year a public lecture by a prominent scientist in the field of cognition, perception cognition, was a good idea, was a good way of celebrating the founder of the local School of Experimental Psychology. And over the years, we had the pleasure to invite and have here, often in this auditorium, really prominent scientists in different areas, not only in perception, the domain of studies in which Kanizsa was mostly active, but also in related areas. As you will see this year, the format will be slightly different because of restrictions, but the spirit is the same as the one from the beginning. So to offer to a large audience the opportunity to know about a specific field, but also to encounter directly a scientist with his own or her own motivations and specific interests and peculiarities. We had the pleasure this year to include the Kanizsa lecture in the festival, Science in the City Festival, of a larger event having Trieste as the focus, Trieste as a city of science. It is a pleasure to include this event, this public event in this festival, because I think the whole festival has the same goal of unifying different aspects of science and culture, and to offer to a very large, the largest possible audience, these ideas. The Kanizsa lecture is organized by Paolo Bernardis, Carlo Fantoni, and myself in the last years. And I will tell you more about the speaker, today's speaker, later, after a brief speech by the president of the local municipal council. Please, prego.
Buongiorno a tutti, mi presento, capisco l'inglese ma non lo parlo. Io sono il presidente del Consiglio Comunale di Trieste, rappresento tutta la parte politica di questa città. Vi porto il saluto del sindaco di Trieste, che per motivi istituzionali non è potuto essere presente in questa occasione. Infatti ne abbiamo parlato questa mattina e mi ha pregato se potevo essere io presente al suo posto. Bene, mi ha colpito molto il titolo che avete dato a questo evento, Pulcini e bambini, come costruire un cervello sociale. Bene, sono molto curioso di sapere come si svilupperà questo incontro, questo approfondimento. Perciò lo ascolterò attentamente e alla fine mi capirò di cosa si tratta, perché non ho capito bene i contenuti. Grazie ancora e vi auguro buon lavoro e buon ascolto. Grazie.
Okay. So also to answer this curiosity about chicks and babies, let me introduce Giorgio Vallortigara. First of all, I must say that I am especially grateful to Giorgio for having accepted our invitation this year, you will understand that there are special reasons for being grateful to him. Giorgio graduated in Padua in 1983, and I have a personal memory of that period because I was there as a full professor, a very young full professor, and I remember Mario Zanforlin, his mentor, Giorgio's mentor, introducing myself to the place, to Piazza Capitaniato, the institute in Piazza Capitaniato. I had a small office, and in a corner there was the chair of Vittorio Benussi. This impressed me very much. And he also told me that there were other colleagues who were in different places, and upstairs there was an office with, and a lab with Giorgio Vallortigara. And that was the first time I think I knew about Giorgio working with chicks in a strange place, for me at least, in Piazza Capitaniato in Padua. And after that period, when he graduated, Giorgio also completed his PhD program in Padua in experimental psychology, having neurobiology as a related field. Then he went to Sussex, at the University of Sussex, as a postdoc, and moving from experimental psychology to neurobiology in a very free way, I would say, and productive way. Then he was a researcher or research assistant, that's just a matter of terminology, in Udine, at the University of Udine for some years. And finally he became, when he became an associate professor, he came to Trieste, and he was in our group, in our faculty for 8 long years as an associate professor, and then as a full professor, becoming also the head of department briefly and dean of the faculty of psychology. I would say that despite that, he was enthusiastic in accepting our invitation. That's remarkable, I would say. Then he moved to Trento, and in particular in Rovereto, where he is very active in a lab in animal cognition and brain studies, comparative brain studies. He is a leading figure in the field. He published in many related areas, collecting an impressive number of citations, more than 10,000 citations in the last 10 years, and more than 2,000 in the whole period. So he is widely known as a leader in research on lateralization in different species. He has been capable of demonstrating for the first time the lateralization of recognition, high-level recognition in avian species, and for instance, and he is open to, as you will see, to different ways of looking at relationships between different bipeds, humans included. The format of this year lecture is rather peculiar, and I will explain you immediately. We have Giorgio here, but soon you will see a dialogue, an interview that has been recorded in the last weeks between Giorgio and myself as an interviewer. And after that, which is the part devoted to, let's say, the biography and scientific biography of Giorgio Vallortigara, and after that we will have a scientific presentation by Giorgio, followed by a public discussion. So I invite you to participate in the discussion that will be coordinated by Raffaella Rumiati here and myself at the end. Lastly, I will mention that at the end, if there will be time, you will have the opportunity to visit the exhibit on the 5th floor on Kanizsa as scientist and painter. The exhibit is called I miei pupoli, sorry, my pupoli, I miei pupoli, which is a familiar term in the local community to label different kind of images used as stimuli in scientific experiments, or also as simple demonstrations or as artworks. So thanks for your attention. Thanks to Giorgio again, and I think that we can move to the interview now. Okay?
I'm going to sit there.
Yes.
Giorgio, your research is highly interdisciplinary, but what do you prefer? To be introduced as a cognitive neuroscientist, a comparative psychologist, an ethologist, or something else?
I would be rather inclined to escape the question by inventing some new disciplinary combination. For instance, I am a comparative neuropsychoethologist. More seriously, Karl Popper once said that a scientist is a student of problems, not disciplines. And in order to solve problems, I think that a scientist must use all the tools, conceptual and technological, that are available. So at the beginning of my career, for example, the use of behavioral methods was dominant, was prominent in my lab. And then gradually a variety of other biological tools were added. So today my lab has every kind of fantastic, fancy toy to explore the animal mind, from molecular biology to single-cell recording to immediate early gene expression. And of course, this is a lot of fun. But obviously, disciplines are historical aggregation of people who share interest for particular problems. But modern neuroscience is interdisciplinary by its nature. So personally, I am not simply a supporter of multidisciplinarity. I am interested in new discipline, in new disciplinarity.
At the beginning of your career, you published in two fields, animal psychology and human perception. What motivated the coexistence of such research interests?
I remember that when I was a student in Mario Zanforlin lab, the option between studying humans or animals was mainly related to costs. Research with animals was, and even today, is more expensive. So when there were no funds for research with chicks, we turned to study perception in humans. And obviously students offer several advantages as experimental subjects compared to other animals. You don't have to feed them, you don't have to clean their cages. Usually they clean themselves. So today the coexistence of these two research interests is continuing quite happily in my lab. For instance, in my current interest for animacy perception, which is the topic of I'm going to discuss in the Kanizsa lecture. The only difference is that today I am more interested in human newborns rather than human adults with respect to visual perception, in order to provide a better parallel to research with newly hatched chicks. As a young student, I remember that I was impressed by the textbook by Heinz Werner, who claimed that comparative psychology should be not limited to investigation of other species, but should include research on development, that is, children, infants, and should include research on people called at the time primitive people, that is, traditional society, the realm of cognitive anthropology, and people with mental problems, namely the realm of psychopathology. I am quite happy because I feel I am close to be an accomplished comparative psychology, because I studied several different animal species. I investigated the cognition in human newborns, and I did also a little bit of cognitive anthropology, because with the late Vittorio Girotto, we did some work on Mayan indigenous groups living in remote areas of Guatemala, studying intuitive notion of probability, and I hope to be able to study with Pierre Picard, Munduruku people, an Amazonian tribe, and the Himba of Namibia with Serge Kasparos with regard the investigation of the so-called mental number line. As to psychopathology, I must confess I miss it as of yet.
Your mentor has been Mario Zanforlin, whose mentor has been Fabio Metelli, a close friend of Gaetano Kanizsa. Please tell us about your relationships with these two grand figures of experimental psychology in Padua and Trieste, Metelli and Kanizsa, respectively.
Fabio Metelli was already retired when I was a student in Mario's Zanforlin lab, but he every now and then came to the institute, which at the time was still called the Institute of Experimental Psychology in Padua. He was a very kind and elegant man, and with other students I had the honor of raising the coffin of Professor Metelli in the ancient courtyard of the Bo, the Cortile Antico at the University of Padua, as part of the ancient funeral ceremony with academic rite. And Metelli behaved with Mario Zanforlin exactly in the same way as Mario behaved with me, and I hope to have behaved similarly with my students, namely leaving and promoting the possibility for young scientists to follow their own interest. Mario was interested in ethology, and Metelli obviously was not a zoologist, so he suggested Mario he actually encouraged Mario to go to Edinburgh and to take a PhD with Aubrey Manning, who was a very eminent, very famous ethologist, a pupil of Nico Tinbergen, the Nobel Prize. And in a similar way, when I started to feel too constrained in behavioral studies and eager to study what happens within the black box inside the brain. Mario, who felt he has not enough competence in neurobiology, suggested me to move to Sussex University and to join Richard Andrews' group. Interestingly, Richard Andrew was also a second generation pupil of Nico Tinbergen, but he was mostly interested in neurophysiology. He was already very famous because of his work on deep intracranial stimulation in the encephalic regions of the chicken brain to evoke innate calls, and interested also in brain asymmetry and memory consolidation. So I went there, and that's how my neuroscientist career began. Cesare Musatti, who had been a pupil of Vittorio Benussi and the teacher, the professor of Fabio Metelli, before leaving experimental psychology turned into psychoanalysis, wrote a beautiful short novel titled Chess and Parricide, in which he talks about the small crime that is perpetrated every time by the student, by the pupil, when he, so to speak, kills his teacher. And true teachers, true masters, know that the best students are the ones who will kill you sooner or later. Well, kill and parricide is a little bit exaggerated, but let's say pupils move into new territories that their masters weren't interested in. As to Gaetano Kanizsa, I met him at the traditional meetings on May, on perception, that the colleagues of the University of Trieste were used to organize. And of course they continued when I was part of them in the period in which I have been a professor at the University of Trieste before moving to Trento. Kanizsa was, of course, a legendary character for us young scientists. And we had some conversation because at the time he published a paper on amodal completion, recognition of partly occluded objects in mice. And he confessed me candidly that the reviewers were puzzled in a first version of the paper because in the discussion he was referring to the animals sometimes with the word rats and sometimes with the word mice. He was not a zoologist, of course, and in colloquial language, rats and mice are very much the same things for us, but actually they are very different species. And Paolo Renzi was co-authored the paper, probably didn't notice during the initial version of the paper the mistake in the discussion. So I was very interested in amodal completion because I was looking for a procedure to demonstrate amodal completion in animals more ecological than the classical procedure of operant conditioning. And more importantly, I was also interested in the possibility to document whether perception of partly occluded objects could occur irrespective of experience. That is, if it could occur in very young, newly hatched chicks, for instance. So I thought that imprinting could provide an interesting model to investigate the problem, because in a natural environment, the mother hen is sometimes partly occluded to the vision of a chick because of grass, because of shrubs. And so with Lucia Regolin, we performed some experiment in which chicks were imprinted on an artificial object, like a red triangle, that became their mother. And then they were tested for choice between an amputated and a partly occluded version of the triangle of the mother. And we did several controls, of course, and we showed that chicks were in fact able to show soon after hatching recognition of partly occluded objects. Then Stephen Lee and colleagues at Exeter duplicated our results with chicks, but using a procedure that was more similar to the procedure in use with human infants, developed by Liz Pelkey and Philip Kellman. And the procedure is shown in this image. There is a rod moving to and fro, partly occluded by an object, and when the occluded object is removed, infants are presented either with an amputated rod or with a completed rod, and fixation time is used as a measure of surprise of the infant. This, by the way, promoted a friendship and a collaboration with Liz Pelkey, who has been also a Kanizsa lecturer and some years ago, and a visiting of my lab, both in Trieste and in Rovereto at the University of Trento.
In a chapter of yours on visual cognition and representation in birds and primates, you asked whether such animals experience an object-filled world like our own. What did you mean exactly by this question, and what is your present answer?
Of course, the windows to the world, the prisms in a sense, that is the various Umwelt of the animals, as von Uexküll called them, differ because different species have different receptor filters. So birds, such as my beloved chicks, for example, with their pentachromatic receptor system, perceive colors that we cannot imagine. However, the interesting question is whether within the limits of the established windows of sensitivity of their receptor of a particular animal species, the continuum of physical stimulation in animals is segregated into biologically significant units, what we call objects, following the same universal rules. This is why it is so interesting to look at visual illusions in animals, such as the famous Kanizsa triangle, a phenomenon which is actually observed in a range of species, from birds to arthropods like insects, for instance.
You have been able to demonstrate that domestic hens and humans disambiguate depth in the same way, using configurations similar to those that Kanizsa used in his work on the impossible experiment. 20 years after the Vallortigara and Tommasi comment on Shepard's perceptual universals, what is your opinion on the basis of perceptual preferences such as the Peters rule?
This is a galliform version of the Peters rule. You can see the image. And the question is the hen, the chicken, behind or in front of the fence? Well, it depends. In the region of the legs, where surfaces have different colors, it is quite obvious that the chicken is behind the fence. But in the region of the trunk, where surfaces are chromatically homogeneous, the hen seems to be in front of the fence, because, according to the Peters rule, this solution allows the visual system to minimize the formation of modal contours. Gaetano Kanizsa made use of Peters rules in some of his paintings to produce wonderful visual paradoxes, and you can see some of them at the exhibition at the Museo Revoltella these days. I believe that these phenomena, which are apparently universal or widespread, at least among vertebrates, we don't know about arthropods, such as insects, for instance, tell us something really important, namely that there are constraints related to the statistical and physical properties of the world that must have been incorporated into the design of any efficient biological visual system. And this is, of course, congruent with the Konrad Lorenz idea, that was before an idea of Wolfgang Goethe, that knowledge is the representation of aspects of an organism environment in that organism body. That is, the laws of aerodynamics for Lorenz are represented in the shape of birds' wings, while the laws of optics are represented in their eyes. This means that there are limitations, probably, boundaries to the way in which not just cognitive system, but also the brains can build up. You can see in this picture some of the brains that we were used to collect with Paolo Zucca when I was here in Trieste. On the top you can see a dolphin brain, and below there are a series of avian bird brains. Consider, for example, the issue of whether other vertebrate species have, like mammals, a prefrontal cortex. We know that in the avian brain there is a region which is shown in this picture, which is called NCL, Nidopallium caudolaterale, which is located in a completely different position with respect to the mammalian prefrontal cortex, and has probably a completely different evolutionary origin. But if you look at the connectivity of the area, that is primary, secondary area that projects to NCL, and if you look at the dense dopaminergic input coming in from basal ganglia to NCL, and if you look at the properties of the cells that respond during the delay of a stimulus in this part of the brain, you can see that the same features are observed. So in short, the lesson seems to be that there are only a limited number of ways to build up a brain. So if you, natural selection, want to do a prefrontal cortex, you will always do it in the same way, either a mammalian or an avian brain.
Along the years, you moved your interests from perceptual phenomena to more abstract processes, like those involved, for instance, in number cognition. You also moved your research from your beloved chicks to other animals like zebrafish and honeybees. I'm wondering what strategy guides a comparative scientist in the selection of an animal model, and how is this choice related to specific scientific interests?
So the distinguished biochemist Hans Krebs once remarked that God has provided biologists with the right organism with which to study any particular biological problem. So I believe that young domestic chicks, for instance, you can see a nice picture of a newly hatched chicks here, are ideal organism, God organism to investigate the problem of the origins of knowledge. Why? Well, because they combine the possibility of a nearly perfect control on past experience, what is going on in the egg and soon after hatching, with the possibility to perform quite sophisticated cognitive tests because they are precocial from the sensory and motoric point of view soon after hatching. Other models can be useful for different reasons. For instance, we are now using zebrafish to investigate numerical cognition. And we have developed behavioral methods. You can see an example in this movie in which zebrafish are trained to discriminate different numerousness. For instance, in this case, you can see that there is an open and a closed door, and the zebrafish is learning that there is a numerosity which is correct, associated with the possibility to exit, and another which is wrong because the door is closed. Or we are also using archerfish. Archerfish are interesting because they spit a jet of water at its prey to use this behavior for number discrimination. And this is shown here in this video, in which you can see that the fish is trained to discriminate between two different numerosity. And in this case, the archerfish has to spit a jet of water on number 3 in order to obtain the reward. And the advantage of these models is that, for instance, using larval form of zebrafish, we can make in vivo calcium imaging of the overall brain and look at which area of the brain are activated during number discrimination. And this is the work we are currently doing with Scott Fraser at University of California, Los Angeles. Or we can also produce genetic lines of zebrafish in which a particular gene is silenced using editing genetic techniques like CRISPR-Cas9, and then look at the behavior of these modified genetic lines in number discrimination tasks.
Let us have a look at the future now. What is going to happen in your lab, and what are your research plans?
Actually, several things. With a postdoc of mine, Elena Lorenzi, and colleagues at Bochum University, Mehdi Beruzi and Onur Gunturkum, we are preparing an atlas of the chick brain, that I consider obviously the chick brain as the apex of the creation, for fMRI studies. So you can see this movie, the overall brain of a chick. And we are also using honeybees to investigate whether a cross-dimensional transfer between discrete numerical and continuous spatial size dimension can be shown in invertebrates, thus supporting the hypothesis of a cognitive universality of a coding for general magnitude. And the availability of this new tracking method allows us to consider another idea, namely whether there is brain asymmetry during the dance of honeybees. So we are using these new machine learning tracking methods to check whether antennal stimulation of a honeybee during the dance is lateralized to one or other brain hemisphere. Or again, I'm coming back to my original interest for memory consolidation process that I studied more than 30 years ago with Richard Andrew. We discovered at the time that with Leslie Rogers that during consolidation, memory is moving from one side of the brain to the other, from one hemisphere to the other. So we are wondering this consolidation process is associated with sleep. And we are investigating this in honeybees, in which sleep can be investigated looking at the movement of the antenna that tend to disappear during sleep state. And this movement of the antenna can be bilateral, biantennal, or just with one antenna only, indicating a selective activation of just one side of the brain. So there is really a lot of fun in science, and I think this is a positive message for the young generation of scientists. To conclude, I would like to mention a citizen of Trieste, Giorgio Dendi, who is an expert on mathematical games and anagrams. And Giorgio Dendi, during a science festival, developed an anagram for my name, Giorgio Vallortigara, and the anagram in Italian is Oggi vorrà alligatori, which means today he will want alligators. Indeed, I still miss alligators in my lab.
Can you hear me?
Yes, Giorgio. You can start whenever you want.
Okay, thank you. So, hope you can hear me. Good afternoon, everybody. First of all, I would like to thank the organizer of this for this kind invitation. So thank you very much to Paolo, Bernardis, Carlo Fantoni, and Gerbino, and to all the colleagues of the Department of Life Sciences of the University of Trieste, of course, and in general the scientific community of Trieste, especially the neuroscientific community. Thank you also to Raffaella, the sister, to be here today. It is a great honor and a great pleasure for me to not only to be part of this obviously prestigious series of lectures, but also for the bonds of affection that bind me to Trieste. As Walter said, a very significant part of my scientific professional life, and not only professional, took place here when I was a professor at the University of Trieste. And here I left many good friends to whom professional esteem and affection bind me. So thank you very much for this invitation. The topic I'm going to discuss today is deeply rooted into the tradition of ethology. And I can show you an historical movie showing Konrad Lorenz with his birds walking and showing the classical following responses by his cheese, and then also swimming in the Danube. Yes, you are familiar with the phenomenon of imprinting. It is a learning process by which the young of some species, usually of precocial species, learn to recognize their mother or their social partner by simply being exposed to the object for a very short period of time, soon after hatching. And in the laboratory, typically the imprinting is studied with young chicks, which are presented with artificial objects, such as a rotating red box. And the chicks are happy to run the equivalent of kilometers in order to enjoy this artificial mother. And you can notice in a minute that if a novel object is presented after initial imprinting, say, for instance, a blue cylinder, going to appear in a moment, the chick would refuse to show any following and any approach response. So as a result of the exposure learning, there is the formation of a recognition memory of the object. And we know what is the area of the brain which is involved in this ability to form imprinting. It is this small semilunar region that you can see here. It is indicated with the acronym AM, which means the medial and intermediate part of the mesopyle, which is basically an associative area, equivalent homologous actually of some cortical region in the mammalian brain. And obviously, if you lesion this region, chicks become amnesic. They seem to be unable to recognize the imprinted object. And there are neurons in the AMM that respond selectively to the appearance of an imprinting object, but not to the appearance of non-imprinting objects. For a long time, people, and particularly Gerbino's groups and his colleagues, were convinced that imprinting chips on artificial objects or any kind of more naturalistic object wouldn't make any difference. But then, looking into a series of several data from several different experiments, something very disturbing appeared. Namely that when chicks were imprinted on a naturalistic stimulus, like for instance a stuffed hen, is not actually a domestic hen, is a jungle fowl, so the ancestor of the species, then a preference for the imprinting object was observed even when the animals were lesioned in the AMM region. So that was very disappointing. In the lab, of course, there was they were afraid that there was something wrong in the localization of the brain region. But then, when looking at the response of chicks when they were presented in the absence of any imprinting, that is, completely naive chicks exposed for the very first time in life to either an artificial object, a simple rotating red box, or a stuffed jungle fowl, they exhibit a spontaneous, inborn, predisposed preference for the hen, as shown here. So Mark Johnson and Gerbino started some work to, in order to understand what is special about the appearance of a hen, and they used several disarticulated versions of the stuffed hen, as shown here, so changing the shape, the silhouette, for instance, in different ways. But this was apparently not making any difference. So there was not any preference for the canonical and the disassembled version of the hen. And the same was true even if large portion of the hens maintaining cluster of features were simply attached to a rotating box. Still, this very weird, strange pattern is not in any way less preferred than the canonical version. But if the cluster is destroyed, and if the single features of the head and neck region, say the eyes, the beak, and so on, are displaced at random in this box, then at this point, for the first time, a preference for the canonical chicken was apparent. So there is something special about the head and neck region, and this seems to be true also for species which are different from the chicken, for instance, a duck, and most surprisingly also for animals which are predators of chickens, such as, for instance, ferrets, providing that they have a head and neck region. Then Mark Johnson moved to study young infants with John Morton, and he discovered that human newborns, newborns of our species, are also predisposed to attend to face-like stimuli a few minutes after birth. They prefer looking by moving their eyes to a canonical face-like stimulus like this one, rather than a completely empty oval, or, for instance, a stimulus in which there is the same features but completely rearranged. More or less in the same years, I was at Sussex University dealing and dwelling with similar problems. I was but from a different original training, that of a perceptual psychologist. I was interested in the fact that textbooks in ethology usually say that any kind of stimulus is good in order to obtain filial imprinting, but better if the stimulus is in motion. So I wondered, is any kind of motion identically effective, or maybe chicks are predisposed to the quite peculiar kind of movement that characterizes a chicken, which is partly illusory, as you know, probably. The problem is that it is not so easy to disentangle the purely kinematic aspects of movement from the shape, the silhouette of the organism. So take advantage of my training as a perceptual psychologist, then I decided to use point light display, which were introduced originally by Gunnar Johansson. These are stimuli in which point of light are located at the joints of the articulation. And if you look at the stimulus as a stationary stimulus, it is not so easy to recognize any shape. But when it is in motion, you can recognize the structure from motion. And even more than the structure, you can recognize the kind of activity the organism is involved with. So we use this kind of stimulus with completely naive chicks coming out from the incubator in the dark, no experience of the world at all. They were placed in a long runway, and on the opposite ends of the runway, there was a monitor screen, high frequency, with a certain number of dots identically, same average velocity, and we compare different kind of motion, like for instance, a walking hand, or rigidly moving, rotating hand. So obviously you can recognize the shape in this case, but the kind of motion is different. It's not the biological motion. Or for instance, we have a variety of control conditions like completely random motion, Brownian motion with constraints at the borders. Or this is quite interestingly, this scramble hand. The scramble is obtained simply by maintaining the local vector of movement of each dot, but displacing spatially. So the neat result is that you can recognize a chicken here, but probably you have the impression, at least from the anthropomorphic point of view, that there is some sort of strange biological creature. So what we found very briefly was that yes, young naive chicks have a preference for the hand walking with respect to rigid motion and random motion, but there was no any difference between the hand walking and the scramble hang. So, and the scramble hang was preferred to rigid motion and random motion. So my original conjecture was wrong. In fact, yes, chicks have a preference, but it is not specific of the movement of the species. They prefer the walking hand to rigid and random motion, but not to a scramble hand. So we turn to a different kind of creature like this one, that is a moving cat. And what we found was that the cat was preferred to rigid and random motion, but there was no difference between a walking cat and a walking hand. So surprise, surprise, chicks have a preference, but it is sort of rough preference in which they are completely happy to approach both a moving walking hand, but also a walking cat. In retrospect, it is not so surprising because consider the situation. They have a learning mechanism, and the learning mechanism in printing would be completely useless if you provide from the start, from the onset of life, all the information about what you want to learn. But what is useful for a learning mechanism is to possess some sort of guidance, some sort of canalization mechanism that would make more quick, more fast the learning process. So imagine natural selection, consider the situation from the point of view of a young chick coming out from an egg. There is several things which are moving in the environment. So natural selection, in order to canalize in printing, the learning mechanism would say to the chick something like that. Chick, please ignore at one extreme completely random motion, say leaves moved by the wind. And on the other side, ignore completely rigid motion like rolling stones. These are not mothers. But in between, there are interesting things to which your attention should be directed, namely semi-rigid motion. Obviously, it is a very roughly defined structure pattern for the movement of biological organisms, because obviously biological motion is common to a walking hand and a walking cat. So if you are an unlucky chick and there is a cat waiting for you when you are exiting from the egg, you are a dead chick, but this doesn't matter from the point of view of natural selection. Okay, you can say, that's an interesting and nice ethological story, probably has nothing to do with our species, with the human species, and that will be not surprise, obviously, because we know that we are phylogenetically quite unrelated. The last common ancestor here of mammals and birds dates back to about 300 millions of years ago. But in fact, this is not so, because in Padua, Francesca Simeon decided to duplicate our experiments using as subjects young human newborns rather than chicks, but using exactly the same stimuli we were using with chicks, so no human figures, but walking hands, scramble hand, and so on. Okay, you can say that's for the Italian part of the audience that there is a bias, probably because these were Paduan neonates, so with some sort of possible interest for chickens, but more seriously, there is no reason to imagine that a few hours after birth they have any sort of knowledge of the kind of motion that would characterize a chicken motion. But nonetheless, they show a preference for biological motion exactly similar to the one we documented with our young chicks. So we turn it back to the issue of the head and neck region on young chicks. Obviously, you can say chicks have no face, but not exactly. If you do some filtering of the high spatial frequency, the pattern which is characteristics in binocular vision for a chick is characterized by the typical inverted triangle with three blobs arranged as an inverted triangle, which has been shown to be so important for infants, for human newborns. So we tested completely naive chicks for preferences for different kind of face-like stimuli, this one, and we found that measuring first stimulus approach or even time spent close to one or the other of the stimulus, they have exactly the same kind of preference which has been documented in human newborns. With Mark Johnson, we actually performed an experiment comparing directly and simultaneously with exactly the same stimuli, human newborns and newly hatched chicks. In this case, we use naturalistic stimuli or white noise, that is stimuli which are controlled for spatial frequency, colors, luminance, average, and so on. And newborns and chicks show exactly the same kind of oriented responses toward the face-like stimulus. Obviously not exactly the same kind of response, because chicks can move, can walk, because they are precocious species. Newborns, we can use only fixation time and preferences in looking time as a measure. So this promoted the starting of a search that has been occupying my lab for several years for this idea. It could be that there is in the brain of all young vertebrates some sort of natural born animacy detector, that is mechanism to recognize the presence of living things, mainly for social purposes, that is for recognition of social partners, but as I mentioned later on, even for different kind of animated creatures. So I summarized briefly what we found on the behavioral side before moving to the more neurobiological part. You know probably that Aristotle has a clear idea about what is special about animated creatures, about what is animated for this inert physical object, namely self-propulsion. The fact that animated things are able to move by themselves, whereas typically a physical inert object can only move if there is some sort of collision with another object. So we decided to do an experiment for imprinting, in this case, reproducing the condition for the classical launch effect studied by Michaud, and then looking whether chicks have any preference for one or other of the two actors, the players of the imprinting sequence, obviously counterbalancing for color, direction, and so on. So this is the classical launching effect you are probably familiar with. There's one object which is self-propelled, and then the second object is seems to move as a result of the physical causation produced by the first object. So very likely the first one could be animated. The second one, well, maybe, but maybe not. It could be simply an unanimated object. Obviously, several controls are needed. For instance, it could be that chicks have a preference for the object which is moving first. So we check for that by changing the kinematic aspect and moving first the second object and then the second. This is good, but obviously there is no contact in this case. So another condition was similar to this one, in which the object is moving and touch, and then there is a delay. And the presence of a delay, as shown originally by Michaud, is enough to abolish the impression of a physical causality. So at this point, it seems that there is psychological causation, and even that the second object is moving as self-propelled, so to speak. Then we have another more complicated control condition to disentangle between the causal action of a stimulus versus the self-propellence. So in this case, the object is appearing from behind a screen in such a way there is uncertainty about whether it is self-propelled or not, but there is physical contact, so physical causation. So briefly, what we found was that only in the first condition there was a preference in chicks, which means that the first object to move is not relevant. The object initiating contact is not relevant. The object having a causal role is not important. What is crucial is that chicks are attracted by objects with, which are self-propelled. That is, that they have psychologically sort of internal energy source, which is what we perceive in animated creatures. Then with Francesca Simeon, we did the same experiment, similar, with human newborns, presenting a sequence with a self-propelled event, like this one, or one very similar in which, because of disocclusion and the starting point, there was uncertainty about whether it was self-propelled or not. And human newborns show indeed a small but quite clear preference for the self-propelled event. Then we move to other kind of signals in the stimuli, which are used as cues for animacy. For instance, changing speed. Changing speed is very effective, at least in human adults, and we check whether naive chicks are spontaneously attracted without any previous experience, visual naive chicks, to objects that change their speed. So we compare very simple stimulus on a monitor screen, moving to and fro with constant speed, with stimuli moving with changes in speed that to human adults are rated as more animated than others. And in fact, we found a strong preference for the change speed stimulus. You can say, well, obviously it is in some way more novel. There is a novelty in the stimulus which is attracting the animal. Yes, but the point is whether novelty in itself is important, or what is crucial is the very moment in which you can observe the change as a perceptual cue, the changing speed. So for instance, if there is a non-visible change of speed, so there is novelty in the sense that the velocity is changing, but you have no experience of the very moment in which this is occurring, then again, there was a preference for the change in speed condition. Or we did a series of experiments in which we have a series of occluders along the trajectory of the object in such a way you can see the change in motion either in the disoccluded part of the trajectory or when it was not visible. And this was enough to abolish or maintain the preference in the young chicks. Finally, more recently, Ursula Rosa Salva with Nikolai Hernik investigated the role of the alignment in the elongation of the body with respect to the direction of movement. That's very important, an MSQ for bilaterian animals, as most of the animals living today on the earth. So we compare, for instance, conditions in which a stimulus is moving, maintaining during motion the direction of elongation along the direction of movement, or different condition in which the object is moving, but at the same time, the orientation is maintained in such a way that sometimes the object is moving in a direction which is not equivalent to the direction of elongation. Again, we have several control conditions for that, but I show you just an example of the data and the distribution. You can see that there is quite a strong preference for this sort of axis alignment with respect to the direction of motion. So the next step was obviously to look at what is going on in the brain that can explain or can account or can localize as a first step the presence of these animacy detectors, life detectors. Ideally, it would be good to investigate this directly in human newborns. And we did some work recently using EEG with Marco Buiatti and Manuela Piazza. We presented human newborns with either a face-like stimulus or an inverted upside-down face-like, or with this stimulus, which is indicated in lab with the nickname of Picasso's face. And we use a technique which is called frequency tagging, which is presenting the stimuli with a certain frequency. This is 8 Hz. It's not so good in the PPT presentation for the smoothness, but just to give you an idea, this is the version with the inverted face. Can show you also the real experiment, and this is the human newborns observing the stimulus. Can show you the face, because this is actually the daughter of one of the postdoc in my lab, so we have the permission to show the face. And frequency tagging allow good signal to noise separation and ratio for the EEG signals. And what we found is a striking signals, you see at 0.8 Hertz for the face-like stimulus with respect to the inverted face-like or the Picasso's-like stimulus. Of course, Marco was also able, taking advantage of some recent neuroanatomical atlases for the brain of newborns, to reconstruct the sources of the signals, which is obviously not so good as for fMRI, but you can see that there is signals in the regions which are the classical fusiform area, more or less. There is also something not very clear to us, this this occipital part, and also this one, which is a little bit of a mystery, but medial activation that we suspect that is mostly subcortical activation from the colliculus. We cannot prove this with the EEG, of course. You can say, well, all right, this is not, however, evidence that this is inborn predisposed mechanism for face-like recognition in human newborns, because differently to the experiment with chicks, in which you have a complete control, accurate control of previous experience, you cannot prevent for human newborns to observe, even very briefly, and have very quick, super fast learning the face of the medical, the doctor or the mom for a short period. Yes, of course, it's quite possible, but consider that we have a certain degree of variability in the age of the subjects, measured in terms of hours. So if you look at the correlation between the age in hours and the strength of the face-like pattern response, you can see that there's a negative correlation, which means that if any effect of experience is there, is that of reducing the preference for the face-like stimulus as the result of the experience with the face-like stimulus, which is, I think, congruent with my idea that this is a sort of supernormal stimulus, similar, take it as an example of other supernormal stimuli which has been studied by ethologists, like the famous begging responses to the pencil colored by red and and white stripes, which is more effective than the a naturalistic head for a gull. Obviously, faces are extremely important for us, even as adults, and you know the phenomena of pareidolia, the tendency that humans exhibit to see face everywhere, in the moon, here, or you can see face also. This is Twin Tower smoke, and people see here the face of the devil, see the eyes and the nose. But my favorite one is the, no, it is interesting to note that the pareidolia is actually not limited to humans. It has been recently observed in monkeys as well. So monkeys prefer to look at faces of monkeys rather than artificial objects. But if artificial objects have pareidolic features, like for instance this little cup or dishwasher, this one, they look longer to this rather than to equivalent non-pareidolic artificial stimuli. Show you this just to show my favorite example of pareidolia, this one. This is a church that is in Florida, in Tampa, and it is the very famous church of the Standard Chicken. Now let's turn to chicks. Obviously, there are severe limitations to the penetration that you can have for the brain using human subjects. So this is one of the reasons we use animal model systems, and chicks are a very good animal model system in this regard. This is the brain of a chick. You can see here the telencephalon. This is the optic tectum, the cerebellum. So very similar to other vertebrate brains. We are not completely blind in our search of the areas involved in the life detectors mechanisms, because in adults of other species, mammalian species, it is very well known that there is a network, so-called social behavior network in mammals that include areas like the lateral septum, the medial preoptic area, and the hypothalamic region as the anterior and the ventromedial hypothalamus, which are important in a variety of social behavior in the adult, for aggression, for gregarious behavior, territorial behavior, sexual behavior, and so on. So we look at particularly to this area, even because they are largely conserved among vertebrates. So basically the same pattern is observed in a mammalian, say a rat or a bird brain with respect to this social neural network. Can I show you more or less the three-dimensional localization of this area in the brain, taking advantage of the fact that we are exploring this also with the fMRI now. This is actually simply MRI. And I can show you an example here. You can see the position of the medial preoptic area, the lateral septum, which are conserved. So they are structures which are observed also in the human brain, and the nucleostenia, which is mostly an amygdalar nuclei. For those who are more interested in the anatomy, this is the location of the septum in an avian brain coronal section. This is here, and there are several subdivisions which are important from the neuroanatomical point of view. This is the arcopalium, it is the homologous of the amygdala in the avian brain. We obviously look also at the AMM that I mentioned before, which is the area in which imprinting takes place, so involved for learning, not for innate inborn predisposition. And as a control condition, we have this medial striatum, which is mainly involved in learning, but not apparently in social behavior. And the techniques we use are based on so-called immediate early-gene expression, which are activity marker of neurons. So basically, the idea is relatively simple. We presented chicks with stimuli that have elements of animacy or that they lack it for a very period of time. in control conditions, say face-like, not face-like stimuli, similar condition, for a period of time short enough to guarantee that there is no learning, no imprinting. Then we sacrifice the kill and we look at the brain, and in particular to the expression of the protein, which is the product of this gene, the CFOS, can be seen by coloring the section, this slice, and then with certain methods we can count automatically the number of cells and see whether there is more activation in particular areas of the brain. So we started with something very, very general, very simple. We wanted to see which brain areas are activated when a chick is looking at a conspecific social pattern with respect to a condition which is looking to nothing. So there is a social pattern for the testing chicks, and the control one is looking to an empty chamber. Olfactory acoustic stimulation is similar, but then there is obviously a lot of difference in terms of visual stimulation. And what we found was that there was two areas that were mostly activated by CFOS, the lateral septum and the arcopalium in general. Then we tried something more detailed. So we presented this time chicks with either an alive chick or a rotating dummy chick, very similar in visual appearance. You can see here an example. But obviously in terms of the pattern of movement, they are very different. Acoustic stimulation is similar, visual configuration is matched, but changes in speed, biological motion, or the dynamic part is deeply different in two conditions. And what we found again is that limited to the right hemisphere, there was a specific activation in the dorsal, but not the ventral part of the septum, and in the preoptic area. Very specific, but not in the arcopalium in general. So we tried something even more detailed, and this time we presented chicks with a very, very simple stimulus. A stimulus in which there is a red disc moving at a constant speed or moving with speed changes. So just 5 minutes of presentation of two stimuli which are very, very similar. And then the result is that there is a specific activation limited to the dorsal part of the septum and limited to the right hemisphere. What about the static feature? So we presented chicks this time either with a canonical staphydang or a scrambled version of it. And again, we look at the brain and we found this time that this region here, more lateral, the nucleus tenea, which is homologous of the medial part of the amygdala, is specifically activated in the brain. So to summarize, quite complicated stuff, probably from the anatomical point of view, if all cues of animacy are combined, there is activation of the septum and arcopalium. If only visual configuration static of the head and neck, only nucleus tenea is activated. If only motion cues are studied, only the septum and the preoptic area seems to be activated. Recently, we are investigating biological motion. This is a little bit complicated. I try to provide you with a simplified version. So we found that if we presented chicks with a walking hand and then presented chicks with the upside down version of the walking hand, they were attracted by both stimuli, but the kinematic was using a different way apparently, because if we measure not approach, but the tendency of chicks to align the direction of apparent movement with the direction of apparent movement of the point light display, they were able to use this information in the upright version, but not with the upside down version. So something like that. If from minute 1 to minute 4, the chicken is moving in this direction, and there is consistencies of chick's body orientation, and when the direction of the chicken is changed, they maintain that. But if they are presented in the point light version, obviously, with the upside down version, they are completely random. The story is even more complicated because when we look at the video recording, we found that this alignment was specific only of the rightward direction. That is when they were using the left eye, so the right hemisphere. And there was also a difference between males and females associated with sex. So recently we looked for C-force expression in several brain regions when chicks were briefly presented with, for instance, rigid motion or biological motion, walking hand. And we found that the nucleosthenia showed no specific activation, but there was a specific difference in different brain regions, in the same brain regions in the preoptic area, in the left hemisphere, in females, and in the right hemisphere, see more activation in the biological motion group in males. So biological motion is complicated because it's modulated in a specific brain area depending on the sex and on the hemisphere of the chick. No activation in the center. You probably know that the social network areas was originally studied in mammalian brain because there is a lot of receptors for hormones in these brain regions. So the obvious question was whether oxytocin, the so-called hormone love, was also acting on our predisposition. Birds have an homologous of the oxytocin, which is called mesotocin. So we injected intracranially mesotocin in the chicken brain, you can see here, and we were able to find that indeed there is an increase in the preference, for instance, for the head region as the result of the administration of mesotocin. Now we are trying to see whether we can abolish by lesion in this area the preferences, and obviously we are also looking at single unit recording. It proved quite difficult to record from the nucleosthenia, for instance, for surgical reasons. We will fix it, but up to now we have some preliminary recording from other areas like the NCL and the mesopallium in general, and we found some preliminary evidence for specific activation in response to face-like with respect to non-face-like stimulus. But this is obviously requiring more work and more cells recorded. Wanted to mention very briefly, without showing details of the data, that animacy detectors obviously are important for social life, but they also refer to other kinds of stimuli, for instance, predators. And we did some work to see whether response to predators is completely inborn, innate, in completely naive chicks. So we presented classical stimuli, a looming stimulus or a sweeping stimulus to young chick. Show you just the movie to provide you with an idea of this sort of experiments. So overhead, the stimulus is presented, which is a looming stimulus, simulating a predator which is approaching the chick and producing a very quick escape response. Quite in contrast, if you simulate a sweeping stimulus, that could be, say, a predator hawk moving overhead, but has not yet noticed you. So the best thing to do is to stop, freezing, and to catch down. You see that immobilize and move down with the body. It is interesting that the immobilization response, from the historical point of view, standing still is not to be noticed. It is a good technique when the predator has not seen you yet, but it can also be useful in the worst case, when the predator has spotted you, and despite your attempt to escape quickly, you are now in its clutches. So many animals react to this situation rather than fighting with the most extreme form of immobility, so pretending to be dead. This reaction is called in ethology tonic immobility, and it is the reaction that gave rise to the various legends about animal hypnosis. And traces of it can be found in this very old book by the Jesuit Father Athanasius Kircher, who described how, by drawing a line on the ground, he was able to hypnotize a hen. In fact, I think that the line is, like other procedure used by magician, as just an ancillary function, because the specific manipulation that induced tonic immobility is to hold back with a little force the hand. So sooner or later, I think we will explore in more detail this very interesting phenomenon. So what we can do with this super chip, what is useful for, apart from, say, basic science? Well, we are exploring two routes currently. One is related to, both routes are related to the possibility that these building blocks of a social brain, if for some reason are not properly functioning at the onset of life, this could be associated with neurodevelopmental disorder, such as, for instance, autism. We are investigating the role of valproic acid. Maybe you are familiar with this drug, which has been used in the past as an anticonvulsivant and a mood stabilizer, until it was found that, unfortunately, is increasing the risk for a mother to have progeny with autistic syndromes. So we injected valproic acid in the egg of young chicks at about 14 days in the embryo. And then we look at the effect on our spontaneous preferences, for instance, for the head and neck region or for speed changes. And we found that there was a complete, the preference was completely abolished in the valproic acid treated animals with respect to the control condition. And interestingly, the imprinting, is the learning, was unaffected. So the chick is able to imprint, say, on a scramble or a canonical chicken, but it has no longer any preference for one kind of stimulus with respect to the other. Consider that this inborn predisposition also have a sensitive period. They are there for a very short period of time, which is different depending on the kind of animacy detectors you are considering. For instance, for change of speed, day one, the first 24 hours are crucial. So for instance, they can be observed, the preference on day one, but no longer is there on day 3. Recently, some colleagues in Japan have been able to identify a substance, which is a thyroid hormone, T3, which is associated with the critical period for imprinting in chicks. And there is a peak of T3 hormone about soon after hatching, when imprinting is occurring. And also, we know about the gene, it's called DO3, which catalyzes the transformation from T4 to T3, and therefore the effect on the imprint. So we wonder whether we can be able to block preferences by blocking T3, thyroid hormone, or also to reopen, and that will be obviously very exciting, the predisposition. So these are our preliminary results, by administering a blocker of T3, which is called IOP, we were able on day one to abolish completely the preference for the change of speed, for the animacy preference. And then we look whether injecting T3, we were able to restore the preference on day 3. Mixed result, because on day 3, sex differences start to appear. And so the results are like this one. There is a reappearance of the predisposition on day 3 in female with respect to the vehicle condition, but not in males. Males seems to show the reverse effect of T3. This is interesting because males also tend to be more explorative and aggressive with respect to social partners. So it could be that T3 is in some way acting in this direction in males. In any case, I think it's terribly interesting that we have some pharmacological agents that could reopen this innate inborn predisposition in the brain. So what about our own species? You know that one problem with children with autism is that the diagnosis is possible only starting from about 2-3 years of age, which is really too late. So it would be very good to have any possibility for early diagnosis of autistic syndromes. In the last years, we organized with the NIDA, which is a network which is has been organized by the Istituto Superiore di Sanità, the National Institute of Health in Italy, to study the develop methods for the early recognition of the disorders of autism spectrum. And they alert us when the siblings of individuals that have been already diagnosed as autistic, they are called high risk for autism, are there all around the country. And so we move with a mobile lab, and we perform on these human newborns, if the parents agree, obviously, exactly the same kind of test we develop for our young chicks. Behavioral tests for the moment, but we hope to be able soon to do also EEG experiments. So these are some of our preliminary data. So the experiments are classical experiment with fixation time. So we presented the human newborns with a biological or non-biological motion, or face-like or non-face-like inverted upside-down face-like stimuli, and this sort of stimuli. What we found, and this is data for newborns which are between 6 and 8 days of age, so very, very early. And you can see these are the results for the low-risk individual, typical development. So they have a preference for the upright face-like stimulus, no preference in the high risk, you can see. And similarly for biological versus random motion, usual preference in the low risk, a typical development. Newborns, no preference, slight tendency for the random actually. And similar results also for biological versus rigid motion. So that's very encouraging. You can say maybe for some probably genetic reason, they lack at the onset of life the mechanism for innate predisposition to face biological motion and other animacy signals. But there is also another possibility that the problem is a delay in development. We have now some initial data for the follow-up at 4 months of age. And there is something very interesting coming in, because you can see that in the low-risk individual, as expected, the preference which is there a few hours, a few days after birth is disappearing at 4 months. This is a well-documented phenomenon. And this is because obviously interest for the individual faces is now coming in with respect to the face-like generic stimuli. But you can see that in the high risk, there is an increase actually. And in some cases, this increase, for instance, preference for direct gaze is also significant. And also for biological motion, you see that there is quite an increase from at about 4 months of age. So it seems that these infants, when they are newborns, are in some way delayed with respect to the appearance of this inborn predisposition for social life. And probably, obviously, this means that they are out of synchrony with respect to the kind of dialogue that they would develop in the normal condition with their mother, which can explain why some of them would develop some sort of autistic phenotype. So I think I can stop at this point saying thank you very much to all my postdocs and mentioning them very briefly. Some of them are no longer in Trento now. Uwe Meyer, Elisabetta Versace, who is now Queen Mary University, Elisa Di Giorgio, who is now in Padua, Matteo Caffidi, Jasmine Loveland, now at the Max Planck in Seewiesen, Elena Lorenzi, Ursula Rosa Salva, and Marco still with me in Rovereto. Marie is moving to England, and Bastien Lemaire still with me in Rovereto. And thank you very much to the ERC for funding my research in this year. Thank you very much, ERC. Thank you very much, Europe. And thank you very much to all of you for your interest and passion for this very long talk. Thank you.
Thank you, Giorgio. And now, I think that it's time for the audience to open the discussion. And if Raffaella wants to moderate the first part.
With pleasure. Giorgio, that was a wonderful talk. Thank you.
Thank you, Raffaella. Thank you.
I really enjoyed it. So it's clearly very exciting to be here to this in this event because first of all because of Kanizsa, and we heard so many good things about him, and we all remember him and his work, but also because of this presentation. So Giorgio has presented a lot of interesting studies, and so if you have questions, curiosities, or whatever, just please raise your hand and ask. I'll give you the mic.
Thank you. First of all, thank you. It was really interesting and enlightening as a talk. I didn't know that there is such complexity in a newborn chick. My question was about the motion of a chicken which is recognized and there's preference. You said that there is more preference for a chicken moving rightwards than leftwards. And if this is, if I understood well, it was something like this. If this is true, can you find confirmation of this also on the adult chicken side as a behavior like chicken moving around their chicks in a clockwise or anti-clockwise way preference in order to make them follow better, or have any sort of things like this?
Yeah,
Thank you for the question.
That's very interesting question. Yes, indeed, there is not just for adult chickens, but for a variety of vertebrate species, including, for instance, dolphins, and of course, the positioning of the children with respect to the mother, the cradling behavior, that suggests that stimulation of the left hemifield for animals that have no laterally placed eyes, like us, for instance, or for animals with largely monocular visual field, like birds, is there for all social behavior. So there is a large amount of evidence from sheep, birds, dolphins, showing that adults tend to prefer the left hemifield to control and to observe, for instance, their progeny, their young, and also to check for their health. Yeah, very general. Consider, however, that in the case of chicks, I'm not saying that they are not recognizing. They have a preference in any case, even for the scramble version. So they have a preference, they approach also the upside down version of the walking hen. What is lacking is the possibility to use the specific kinematic in order to recover the direction of movement. This is lacking as a result of the upside down version. And apparently for this sort of computation, the right hemisphere, the left eye, is particularly important. That could be related to social behavior in general, but it could be that it is something specific in order to process spatiotemporal aspects of the stimulus. That could be also a specialization of the right hemisphere.
Thank you, Giorgio. There is a question from Davide Zoccolan.
Hello, Giorgio. Nice talk. Very nice to see you, although you are far away. So I had a question which is concerning mostly the visual part of what you have shown. So I'm very interested in general in how much the visual system, how much of what the visual system does is innate and how much instead is acquired through experience based on the statistics of the environment, right? And, you know, I'm working with rodents, and in rodents, the story is very complex. Some groups have reported that if you keep rats in the darkness for the whole critical period, then the visual system is basically very much immature. And so if you go and record into visual cortex, you see neurons that have a very poor selectivity. We have done a study recently in which we are seeing that by manipulating the statistics of the environment, we actually can reduce the development of complex cells, which is a very important kind of neuron in V1 in rats. But other studies in mice show that mice seem to be coming out almost with the whole visual system working, right? And here, by looking at this ability of the chicks to discriminate, something that seems quite a complex pattern, because this innate preference that they have for these features of the face or the hand or other body parts is really surprising to me, right? I would expect that in order to be able to integrate into some sort of more or less complex configural representation, this feature, they will need experience for this. So I wonder, how you can put together all these different findings that perhaps are different based on the species, or do you find surprising that your chicks are so good at immediately being able to recognize very complex patterns?
Right. Thank you for the question, Davide. Obviously, well, first of all, I'm not pretty sure that they are complex stuff. Consider, for instance, that for the face-like stimulus, we are looking at a supernormal stimulus, so no real natural faces, but a very pre-prepared version of it, just 3 blobs, high contrast arranged into a surrounding. Consider also that on the mammalian side, there is quite good evidence that in human newborns, I mentioned our results with EEG, for instance, there is clear evidence that the face-like response is there in very, very young human newborns. And it is of today, I think, the paper by Nancy Kanizsa showing that in congenitally blind individuals, the visual recognition, the recognition of face does not require any visual experience. However, with respect to the role of experience, I think that the story is a little bit more complicated than I described, obviously, for the sake of simplicity. So I'm not arguing that there is no role of experience. I am arguing that there is no role of a specific experience, which is quite different. Namely, all the evidence from the early work related to the head and neck region by Gary Lowen and Mark Johnson are shown that experience is important in the sense that it's providing generic sensory experience, is providing sort of permissive role, but not instructive role in the development of this predisposition. For instance, preferences for biological motion require specifically that chicks are stimulated in the dark by allowing them to run into a treadmill. And this motoric activity is also associated with an increase in the level of androgens in the brain. And this is required. I insist to call this predisposition as innate, as inborn, disgusting sometimes also my ethology colleagues, simply because while I recognize the role of experience, I do not recognize a specific role of specific experience. So, of course, you cannot perform the perfect Kaspar-Hauser experiment. Yes, I agree on that. And so there is a permissive role of other kind of source of stimulation. So stimulation, generic stimulation in the visual domain, generic stimulation in the acoustic domain motor activity is needed in order to act as a sort of trigger for all the predisposition I described, for the face-like, for the biological motion, for the speed changes, and so on. But there is no evidence that any specific instruction mechanism is there in order to provide, for instance, structural information for the face. Of course, consider that we are using an cause, they say in French, a precocial species. And obviously this is the reason I selected this particular kind of model organism, because it allowed me at the same time to have very precise control on past specific experiences. I'm pretty sure that my chicks haven't seen any face-like stimulus before the experiment, but at the same time they are mature enough from the point of view of the neuronal responses, for instance. You know, this is a general problem when studying mammalian brain. For instance, consider the evidence in the spatial domain concerning from Neil Barge's lab and from people in Norway lab concerning whether head direction cell, place cells, grid cells are there at the start. Well, to check for that, you have to record, it's very difficult from a technical point of view, rats when they are leaving for the very first time their nest, sort of technical to the force, but providing evidence, for instance, encoding of direction by head direction cell is there before they have experience of surfaces. They have the eyes closed before leaving the nest.
Thank you.
Hi, Giorgio. I'm Giovanni, so you know me, Giovanni Mirabella. Thanks for this very nice talk. I was wondering about the effect that could have on imprinting in chicks the sort of biological motion of predators. So if you have a predator who is showing an aggressive behavior and probably the chicken, instead of having an approaching behavior, should have an avoidance behavior. So you should have a sort of the possibility of cancelling the imprinting. Is this the case?
We don't know. That's a very interesting question. We are planning to investigate whether they are able to recover, to extract more subtle signal than simply moving biological. Similar to the stimuli I show you, showing that you can recognize the sort of action. There is some evidence in a completely different domain. Chicks can be trained to avoid poisoning stimuli, food, by looking at a conspecific that after pecking something, which is disgusting, produce a sort of disgust display, moving the head in this way, try to clean up on the floor, and so on. It seems that the pattern of motion in itself, in this case, is providing information enough to a conspecific in order to avoid, to peck the same object they have seen pecking on the basis of the pattern of movement. So it could be that there is this sort of information available in the biological motion display, but this has not been properly investigated as of yet.
Doesn't seem that the natural selection has done a great job in providing a mechanism that is so generic. So you want to recognize somebody who is, to differentiate somebody who is actually coming against you or somebody who is just walking by or passing by. So my question is, Giorgio, what does determine the selectivity of this mechanism?
The term, sorry?
So what is it that determines the selectivity of this mechanism? Why do you think it's, well, if it say that you do not, you will not demonstrate that is more selected than you have found so far. So why is it that there isn't such a mechanism in chicks?
I think that the reason has mainly to do with improving learning. So basically, I think this is generally true with respect to, and I think the problem with the neural network, deep learning and all this stuff. I mean, I think it is needed to have some sort of biological priors to make all this stuff biologically plausible. And biological plausible means that something that might operate in a natural environment in relatively short time. Yeah. The problem with imprinting and the problem with associative learning in general is that if you imagine that really the brain is a tabula rasa, that there is no priors, then you need a lot of time in order to learn everything. So the raison d'etre of this sort of biological predisposition is basically to make learning possible. This is a very Kantian idea in a sense. Huh?
Yes, indeed. Are there other questions in the audience? So while you prepare the next one, I'm going to ask you another thing very quickly. So towards the end of your presentation, you presented these very interesting and promising results on autism. So my question is your position so far like there is a problem, there might be a problem that is perceptual in nature, and this is going to compromise and say somehow affect the later social or emotional development. Is this the idea?
Yeah, exactly.
Okay, so that the building blocks are perceptual in nature, but then on this, so, and if this goes wrong, then you cannot build up anything more complex.
Right. You are disrupting the social relationship.
Okay, yeah. That, yeah, sounds plausible. What I was I had a curiosity that was this. So aren't you planning to follow up these little babies who they seem to show some difficulties? Because that, I mean, you would like to see whether your predictions are actually, or your findings are confirmed later. Hopefully not. I mean, not for the children at least.
Yes, of course. We are waiting for the final data, so to speak, and they will be about 3 years old, and to see basically how many of them would exhibit a number of the disease. Consider, however, this is typically quite a slippery issue.
I know.
For the people, because they are using this very general and generic term of autism phenotype, which means any sort of things. So you can have apparently very, very healthy, in a sense, people that have very subtle deficit or abnormalities in their social behavior without pathology.
Thank you, Giorgio. So, well, if there are no other, please.
So, I had another question about the autism spectrum. Autism spectrum is quite a complex disease with many symptoms, so that sometimes it's difficult to find a unique biomarker of the disease. But I was interested in your idea because some of the people think that one biomarker of autism is inhibitory control, motor inhibitory control. And what you have shown is that there might be a compromised perception of motion. So could it be that generally autism could arise from some motor problem or interpretation of motion or some cognitive motor problems? So that basically the impairment born from the inability to understand more generally intentionality of others. And this could be a broad thing.
Yeah. That's obviously a very interesting idea. I'm not pretty sure. Obviously, consider, first of all, that in our animal model, we have no evidence for, up to now, for agency. Is most of our work, all of our work up to now has been limited to the province of animacy. And animacy is not the same as agency when you have recognition of intentional behavior by others. And I think this is quite crucial for our species and, as you said, for autism. With respect to a specific role of motion or motoric activity, again, I'm not pretty sure because, for instance, in the case of responses to face-like stimuli, which is one of the more clear data we have, there seems to be no any motoric involvement because we are not using naturalistic face, but we are using face-like stimuli, which are completely motionless, and they seem to be not terribly interested for these high-risk newborns.
I have a question, Giorgio, about the contrast between normal face or other configuration versus upside-down configurations. You used and showed us several results about schematic faces and biological configurations, motion configurations, like hen or point-light walker. But there is a big difference between these two situations, apart from one is static and the other is dynamic. The faces in the upside-down version are not actually upside-down faces, schematic faces, but only the triangle is upside down relative to a contour of the body, of the head, which remains constant. And this is, I think, a problem because you confound orientation with the structure, because the inner structure of the configuration is different in the upside-down schematic face. This is one observation. The other is the role of gravity or the pattern of acceleration, deceleration in the walker, or any human walker or hen walking, whatever. Because when you reverse the orientation, you actually display a different dynamics. If the animal, the perceiver, has some general schema of the gravitational environment, so an environment with a ground. So this is the really interesting question. There is any difference between precocious species and humans, for instance, with respect to the knowledge of gravity, which is revealed by the inversion of the walker, for instance, the point-light walker?
Well, apparently not, in the sense that in our chicks we have got, for the upside-down version of the biological motion, exactly the same kind of response you can get from human subjects. That is an inability to use the kinematic of movement exactly not because of lack of recognition, because in fact they recognize, they are attracted by the stimulus, but because of lack of the proper direction of movement with respect to gravity. So, obviously, the classical evidence by Shipley, in which he has an acrobat film moving on the hand, and it is moving with the correct orientation or in this way. And what is important is not the direction of the orientation, but the proper movement with respect to the direction of gravity. This seems to be observed also in chicks, apparently. And what is interesting is that our chicks, differently than human adults, they don't have any experience of looking at objects moving in a particular direction because of gravity. in a natural world. So it could be that they are using or obtaining this information in the egg, for instance, from movement of their bodies.
Okay, thanks. Yes.
One question from the web.
Okay.
Yes, thank you. The name of the person is Frederick Halper, and the question is, would you expect to find any difference in using profiles or view from when you use point light workers? The point of view is frontal versus lateral. If you expect any difference when you use these two types of stimuli?
We didn't check this with chicks, but I know, for instance, that with pigeons, and recently there has been some work carried out with dogs, suggesting that there is more response, more information apparently conveyed by positioning as a profile rather than in frontal presentation. In chicks, we always use this sort of presentation that I show you, so I don't know for our chicks.
Okay.
But in yes.
Giorgio,
yes.
if you have finished, we would like to thank you for your very interesting, exciting presentation, and also for the interview. And I personally thank the organizers. This is an appointment that is very dear to all of us in Trieste and also outside. So thank you very much for doing it. Thank you.